The B and T lymphocytes are key players in adaptive immunity, our body's third line of defense. These lymphocytes are like soldiers defending a medieval castle. Like archers, B cells can hit faraway targets, but they shoot antibodies, not arrows. Some T cells are like foot soldiers, engaging in hand-to-hand combat with one cell attacking another. Other T cells act like officers and help direct the battle. B and T lymphocytes have different functions in the immune system, but they share some common features. These common features are the focus of this topic. Your goals for learning are to outline the shared features of B and T lymphocytes, to describe the structure, function, and sources of diversity of B and T cell antigen receptors, understand how immune reactions to self-antigens can be avoided, to explain why lymphocyte recirculation is necessary for effective host defense, distinguish between effector cells and memory cells. Here's what you need to know. Key characteristics of adaptive defense mechanisms, the nature of antigens and antigenic determinants, what cell membrane receptors are and how they work, the microscopic anatomy and function of the thymus, the function of primary and secondary lymphoid organs. To see definitions of terms, click the bold red words. The shared features of B and T lymphocyte function include specificity of receptors. B and T cells both have receptors that are specific for a specific antigen. Diversity of receptors. There is a huge number of different lymphocytes, each with its own unique antigen receptor. Regulation of activation. Because of their destructive capabilities, both B and T cells must be tightly regulated. Activation of either B or T cells lead to the formation of a clonal army of defenders, a process called clonal expansion. Click either of the B or T cell soldiers to see clonal expansion. The last shared feature is memory. After an initial encounter with antigen, long-lived B and T cells act as a kind of memory so that when the antigen is encountered again, the immune system can mount a more rapid and effective response. Click any B or T cell soldier to see memory. Let's take a closer look at the targets of the adaptive immune system, antigens. The environment outside the body contains a vast number of antigens, each with multiple antigenic determinants. Likewise, the environment inside the body has a set of normally occurring internal antigens, called self-antigens. The trick for lymphocytes is to recognize antigens from pathogens while ignoring self-antigens. Self-antigens are the shapes that lymphocytes expect to find in the body. Lymphocytes must not mount a defense against these shapes because doing so would lead to autoimmune disease. Click the virus-infected cell to see a T cell recognize its viral antigens and destroy it. The ability to distinguish pathogen from self depends on the specificity of lymphocyte antigen receptors and comes about through a process called education, which we will discuss later in this topic. Everything that the immune system does depends on its ability to recognize certain shapes or antigenic determinants that identify friend and foe. In adaptive immunity, recognition occurs when the target shapes bind to membrane-bound receptors. The surfaces of mature B and T cells are studied with lymphocyte antigen receptors that allow these cells to identify their particular antigenic shape. The antigenic specificity of B and T lymphocytes is determined by the shape of their antigen receptors. Click this B cell to see its lymphocyte antigen receptors. There are two features that you should observe about these two B cells. First, all of the antigen receptors on a given cell are identical. Now click the other B cell. The second feature you should observe is that while the antigen receptors on the two cells have the same general shape, the antigen specificity, as determined by the shape of the ends of the arms of the Y-shaped receptor molecules, is different. Each B or T lymphocyte expresses 10,000 to 100,000 antigen receptors of identical specificity on its plasma membrane. The receptor specificity of each B or T cell is unique, that is, each B or T cell binds optimally to only one antigenic determinant. While the antigen receptors of B and T cells have a related structure and similar function, there are important differences in their shapes and the way in which they interact with antigens. The B-cell antigen receptor is an immunoglobulin or antibody molecule. This antibody molecule is basically identical to secreted antibodies, except that it has an additional segment that extends through the plasma membrane, anchoring the antibody to the cell. The structure of antibody molecules is discussed in Topic 5. Click the B cell to enlarge the B-cell receptors. When antigens occupy the antigen-binding sites on the B-cell receptors, the B-cell receptors signal the cell that antigen has been found and recognized. Click the bacterium to see it bind the receptor. Like B cells, different T cells display T cell receptors specific for different antigens. Click the bacterium to see T cells and their lymphocyte antigen receptors. Unlike the B cell receptor, the T cell receptor is not a membrane-bound antibody, and it does not have the characteristic Y shape of antibodies. Click a T cell receptor. Remember that B-cell immunity is humoral immunity and is directed against antigens free in the extracellular fluid. On the other hand, T-cell or cellular immunity is directed against the body's own cells when they are invaded or have become cancerous. Click the virus-infected cell. T cells identify target cells by the clues on the surfaces of the target cells. These clues are antigen fragments cradled inside special cell membrane proteins. These proteins are called the Major Histocompatibility Complex (MHC) proteins. In Topic 6, we will learn more about how antigens end up on the cell surface bound to MHC proteins. For now, what you need to know is that the T-cell receptor binds to the combination of MHC and antigen fragment. Click the T-cell to see it identify antigen on an infected body cell. As we've just seen, to respond to any antigen, the body must have a lymphocyte with receptors that are specific for that antigen. The problem that the immune system faces is that there are millions of different potential antigen shapes that it may have to recognize. Click the B cell to learn how the immune system solves this problem. Our immune systems make a wide variety of receptors in advance before the antigens for those receptors ever enter the body. When antigen binds and activates one of these receptors, the cell divides repeatedly, making a clone of identical cells with identical receptors. The immune system mechanism of matching receptors and antigens is called clonal selection. It is the antigen that selects the lymphocyte by binding the correct receptor shape thereby causing more copies of it to be made. Drag the antigen to a B cell receptor to select it and generate a clone. This receptor does not match this antigen. Try again. The clonal selection theory requires that the body make a large number of lymphocytes, each with unique antigen receptors, in hopes that one will match whatever antigen might come along. Just how many different cells is that? Click the lymphocyte to get an idea of the number of cells. Think about all the different antigens, each with a unique shape, that your body might ever encounter. This number is potentially huge. In fact, your body makes about 100 million different types of lymphocyte antigen receptor, each just waiting to be selected. As you now know, the immune system must generate about 100 million different kinds of lymphocyte antigen receptors. And as you also know, these receptors are proteins whose structure is encoded by genes. Yet, there are only about 25,000 different genes that make up all of the genes in your body, only a fraction of which code for immune system proteins. How do our bodies generate so many different receptors from so few genes? Click the antigen receptor to find out. The answer is that we only use a few genes, but we mix and match bits of DNA from within those genes to form the final code for the protein, much like the colored blocks of a genetic Lego set. When the immune system builds a given type of antigen receptor, certain parts of that receptor are always the same. These parts are called the constant regions and have the same functions for all receptors, like anchoring the receptor in the membrane and signaling the cell when the receptor is bound. Click a constant region to continue. The parts of the receptor that get the job of binding antigen are variable regions. These are encoded by small chunks of DNA that have been randomly assorted. Click a variable region to label it. In this animation, we use different colors to represent different shapes. For antigen receptors, these randomly chosen different shapes determine the receptor's specificity for antigen. Both B and T cell receptors are built using a similar process, but we will use the T cell receptor as an example. Let's build some T cell receptors by randomly selecting DNA fragments. Click the T cell. Click once on each jar to randomly select DNA. Then click the wizard to create the antigen receptors for your lymphocyte. To make a different receptor for a different lymphocyte, repeat this process. Click the antigen receptor to continue. Building the variable antigen binding region of an antigen receptor is a bit like building a chair by grabbing random pieces of wood out of a lumber pile. The size and shape of the chair depend on just which bits of wood were selected, and sometimes, no matter how hard you try, those pieces of wood won't make a chair. When this happens with receptors, the cell that makes them dies during development. Both B and T lymphocytes originate in the bone marrow. While immature B cells remain to mature in the bone marrow, immature T cells migrate to the thymus and mature there. Because lymphocytes originate and mature in the bone marrow and thymus, these organs are called the primary lymphoid organs. Click the bone marrow to allow immature T cells to migrate to the thymus. In order to become successful mature lymphocytes, B or T cells must accomplish two things. First, each must successfully generate a viable lymphocyte antigen receptor by randomly shuffling its DNA segments. Second, each must survive the education process, a daunting series of practical exams that will result in as many as 19 out of 20 cells self-destructing by apoptosis. Oh my, and you think you have it bad. Click the bone marrow or thymus to allow mature lymphocytes to enter the circulation. Each mature lymphocyte is immunocompetent in that it has a viable lymphocyte antigen receptor and is self-tolerant. It will not react to self-antigens. Click the immature T cell to learn about its education in the thymus. While we focus on T cell education in the thymus because it's better understood, B cell education is thought to be a similar process. Immature T cells migrate from the bone marrow through the blood and enter the thymus. In the outermost cortex just below the capsule, immature T cells undergo a few cell divisions before shuffling their DNA and forming new T cell antigen receptors. Click the immature T cell to generate receptors. These cells now undergo thymic education and testing. During this process, they migrate from the cortex to the medulla. Let's follow a single cell through the testing process. Click the immature T cell to begin. T cells only recognize antigen when it is bound to major histocompatibility complex (MHC) proteins. The first test that our T cell must pass is called positive selection, which requires that its receptor recognize MHC by binding to it. Only cells that can recognize MHC are allowed to mature further. This test is administered by antigen-presenting cells of the thymus, such as the dendritic cell shown here. Drag the immature T cell to the dendritic cell to learn about positive selection. Click the No arrow to see the fate of immature T cells that fail to recognize self-MHC. Cells that fail to recognize MHC die by apoptosis. Click the Yes arrow to see the fate of cells that recognize self-MHC. Cells that recognize self-MHC survive to proceed to the next test. The second test, called negative selection, is designed to protect the body from being attacked by T cells that recognize the body's own antigens. Like the previous test, this test is also administered by antigen-presenting cells. But in this test, antigen-presenting cells will present a great variety of self-antigens to the T cell. Drag the immature T cell to the dendritic cell to learn about negative selection. Click the Yes arrow to see the fate of immature T cells that bind to self-antigens. Immature T cells that recognize self-antigens die by apoptosis. Click the No arrow to see the fate of cells that do not bind to self-antigens. Immature T cells that do not recognize self-antigens are self-tolerant. They have now passed all the tests and are allowed to continue to mature. Negative selection of lymphocytes is as good as it needs to be, but it is not perfect. The immune system allows lymphocytes that react weakly with self-antigen to survive and circulate through the body. This allows the immune system to fight off pathogens that have antigens similar to self-antigens. The resulting problem is that sometimes these self-reactive lymphocytes go ahead and attack the body's own cells, which can lead to autoimmune diseases. See if you can match the targets of the autoimmune response with the diseases listed here. Drag the autoimmune response target to the disease on the easel. Well done! Click the red blood cells. There are several mechanisms whose purpose is to prevent autoimmunity. For those weakly self-reactive lymphocytes that have escaped negative selection, there are two mechanisms that ensure self-tolerance. First, the process of activation of lymphocytes contains built-in safety checks. Second, certain T cells, called regulatory T cells, act to suppress self-reactive lymphocytes. We will discuss both of these in more detail in topics 5 and 6. Normally, these mechanisms are enough to prevent autoimmunity, but sometimes these self-reactive lymphocytes slip out of control. Click each event that you think might cause this loss of control. That's correct. What else could trigger autoimmunity? That's correct. What else could trigger autoimmunity? That's correct. All of these can lead to autoimmune diseases. B and T lymphocytes that have completed their education in the primary lymphoid organs are mature immunocompetent lymphocytes. Because they have not yet encountered their one specific antigen, we call these lymphocytes naive. There are two possible strategies that a naive lymphocyte could use to find its antigen: wait for antigens to come to it, or hunt for their antigen. Select the strategy you think lymphocytes use. Yes, lymphocytes hunt for their antigen by continuously circulating throughout the body, testing for their antigen as they go. This approach increases the likelihood that a rare antigen will meet its equally rare receptor. Click one of our naive graduates to see it circulate. Lymphocytes leave the blood and enter secondary lymphoid organs, such as the spleen and lymph nodes, in search of antigens. If they fail to find antigens in secondary lymphoid organs, then they return to the blood. For example, lymphocytes return to the circulation from lymph nodes by leaving via the efferent lymphatics and returning with the lymph to the venous circulation. Lymphocyte activation occurs when a naive lymphocyte encounters its antigen. This process begins with clonal selection because binding of the antigen selects which lymphocyte becomes activated. As we will see in the next two topics, activation of both B and T cells is tightly regulated and includes a number of safety checkpoints. The repeated cell division that follows lymphocyte activation is called clonal expansion. Activate the T cell and see clonal expansion by dragging it to the dendritic cell and binding the antigen. The repeated cell division that follows the encounter with antigen is clonal expansion. The result is a clonal army of about 1,000 cells, each with identical lymphocyte antigen receptors. Most of this clonal army is made up of effector cells that go off to battle against the invader. The remainder of these cells become memory cells that continue to circulate and can be reactivated if this antigen is ever encountered again. Both B and T cells form effector cells and memory cells, but the effector cells act in different ways. The specific actions of the effector cells produced by B and T cells will be discussed in the next two topics. When an antigen is encountered for the first time, a primary immune response is generated. Let's examine how this response unfolds over time. First, we will introduce an antigen, we'll call it antigen A, into the body. Click the splinter to begin. The antigen usually enters the body across mucosal membranes, but it can also enter the body through the skin, as shown here. Over the next several days, antigen A is carried through the lymphatic vessels to lymph nodes or through the blood to the spleen. As we have seen, the secondary lymphoid organs are the sites where lymphocyte activation occurs. Click the lymph node to begin lymphocyte activation. Following interaction with antigen, the activated lymphocyte undergoes clonal expansion to form an army of effector and memory cells. Click the B cell to expand the clone. In this case, antigen A has activated a B cell, which forms a clone of effector cells called plasma cells. Plasma cells will secrete antibodies that bind and promote clearance of the antigen. Similarly, T cells activated by this antigen would form a clone of effector T cells. Click a plasma cell to begin antibody production. The magnitude of the response to antigen A can be measured most easily by determining the amount of anti-A antibody in the blood. T cell activity, while not as easily measured, follows the same pattern of activation and a similar time course. Click the antibodies to measure their concentration over time. In this example, antibody to antigen A begins to appear in the blood by about day 7. Its concentration rises to a peak for about a week, and then declines as antigen A is cleared from the body. What we've just explored is the primary immune response, which occurs on first exposure to an antigen. Click the graph to rerun the animation as a continuous sequence. You have just seen a primary immune response. If an individual is re-exposed to the same antigen, the memory cells that were created by previous immune responses generate a secondary immune response. Click the splinter to elicit a secondary immune response. The generation of hundreds of memory cells during the primary immune response created a large pool of cells available to respond upon re-exposure to the antigen. The larger number of memory cells means that the cells will find the antigens more quickly and generate a larger number of effector cells each time the antigen enters the body. Thus, secondary immune responses are generated faster and are of greater magnitude than primary responses. Click the graph to compare the two responses. Secondary immune responses are often sufficiently effective that pathogens are cleared before symptoms of an illness appear. This explains why we may get sick from a given pathogen only once. Some pathogens, such as flu viruses, avoid detection by continuously changing their surface antigens. Artificially introducing antigens to the body, a process called vaccination, generates memory cells during a primary immune response, which can then protect us without the inconvenience and risk of getting sick. To see this animation again, click the finger. Here's a summary of what we've covered. Each lymphocyte has multiple identical copies of an antigen receptor, but receptors on different lymphocytes are unique. Antigen receptor diversity is produced by random recombination of gene segments. During development in primary lymphoid organs, lymphocytes become immunocompetent and self-tolerant. Lymphocytes continually circulate throughout the body in search of their one specific antigen. Binding of antigen to a naive lymphocyte's antigen receptor causes clonal selection and clonal expansion, resulting in the production of effector cells and memory cells. Secondary immune responses are faster and of greater magnitude than primary immune responses. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.